trilha super legal, uma trilha nova. A trilha que começa hoje é a trilha de Health and Science. E hoje a gente vai ter uma aula muito especial com a Dr. Denise Foley, da Universidade de Santiago. Ela vai falar um pouquinho com a gente sobre vacinas. Isso é muito legal, um assunto muito relevante hoje, né? Gente, lembrando, final da trilha tem prêmio para quem participou de 3 aulas, hein? E quem não está seguindo a Efígie no Instagram, por favor, segue a Efígie no Instagram. Lá vai ter muita live legal. Lá a gente posta prêmio, posta todos os schedules das aulas que vai ter e tudo mais. Tá? E aqui na tela tem o QR Code do Telegram. Todo mundo que estiver no grupo do Telegram tem sorteio especial, hein, gente? Fica de olho. Ok? Vamos ver quanto tempinho. 2 minutinhos. Vamos dar mais um tempinho para o pessoal entrar, que tem mais gente entrando. E já já a gente começa a aula. O pessoal está entrando. A Ana entrou. Oi, Ana. Então, para quem está entrando agora, que tem bastante gente entrando agora, hoje a gente vai começar a trilha de Health and Science. Hoje a aula com a Dr. Denise Foley, tá? Sobre vacinas. Então, vamos começar a aula? Welcome, Dr. Denise. Thank you so much for being here today.
Thank you for having me. Looking forward to the class. Welcome, everybody. So is this my cue?
Yes, ma'am. Go for it.
Okay. Welcome, everybody. I'm from Santiago Canyon College. As mentioned, we're in Orange, California, so Southern California, and I'm going to be talking today about vaccines, giving you a little bit of background information about the different types of vaccines. This is a very timely topic, right? They're very much in the news today. There are a couple of other people from my campus that are joining me today, and so maybe they can help answer some specific questions later. So we have Esther Mead from the International Office and also Danielle Alared, who is the Career Education Coach. And so they're here to also help answer any questions that you have. So let's get started. I'm going to try to share my screen. Hopefully this is going to work out, and I'm going to pull up my slides right now. So you can see that. Let me see if I can get it to present. My computer is a little bit slow. It doesn't really like all of the zooming, but hopefully this is going to work out. All right, so I'm from the biotech program at Santiago Canyon College, and we're going to talk today a little bit about vaccines, and then in the end, we'll talk a little bit about our program. So just a really brief introduction. There's me in the corner, Denise Foley, and then there's 2 other full-time biotech faculty on our campus, Professor Anson Louis and Professor Novi Bath. And here's Danielle Alared, our career education coach that is also part of our team. And there are some part-time faculty as well. So you might be wondering a little bit about exactly what is biotech, because that's going to be the basis of developing the vaccines. And biotechnology is really just the application of technology to solve problems, to make useful products, and it's based on the study of life, of course. That's the bio part of it. So there are very many opportunities in biotech. Biotech is a very large umbrella term with a lot of other industries and fields that are related to it using the tools of biotechnology, and you can just see a few of them right here. And of course, we're going to be focusing today on this tile up at the top on vaccines and how biotechnology has helped us develop some different types of vaccines. We're going to talk a little bit about that right now. So that's our topic, vaccines, and also another related technology, passive protection. We'll talk a little bit about that at the end and what the difference is, because you might be hearing a little bit about that particular type of treatment for diseases as well. So I just want to, as I'm entering into this, if there are questions, then you might go ahead and unmic yourself and ask a question. I'm perfectly happy to answer them as they might pop into your mind, but you can put them in the chat. I have a little bit of a difficult time seeing the chat and my slides at the same time, so I might not see it right away, and somebody could, read it out to me, or I'll get to it in a little bit. Okay, so just letting you know that questions are welcome. So the term immunization is really about stimulating our immune system to protect us against different diseases. And there are two different ways that we can stimulate our immune system to protect us. The first is active, and the second is passive. So active is really our immune system learning how to respond, and it will remember how to respond to something. And that's really more beneficial to us because it's a long-term prospect, something that we would be able to be protected against for a very long time, maybe possibly even our lifetime, if our immune system learns how to respond. Passive protection is short-lived. It's using somebody else's immune system essentially to help our immune system clear out a protection. We'll talk a little bit more about that later, but it's very short term. It's something for emergency situations in order to help clear an infection that is threatening the life of somebody. So what about immunization? A little bit about the history. When did it come around, right? When did we figure this out? Well, we know that the Chinese, way back in the 12th century, they wrote about how children that had smallpox, which is a very devastating disease that we don't have to worry about anymore because of vaccines, right? Back in the 12th century, Chinese knew that if a child recovered from smallpox, they wouldn't get it again. So that was something that they were very interested in because that was something that was protective, right? And smallpox was devastating. So they instituted a practice called variolation, which could induce immunity. And really what that involved was when you have smallpox, you get scabs that develop on your body that as you're healing from that disease, because you get these pocks all over, right? These lesions on your body. And as you're healing, you get scabs. And what the Chinese would do is they would take the scabs from somebody that was getting over this infection, they'd grind it into a powder, and then they would allow somebody else that was susceptible to the infection, had never had it before, to inhale some of that powder. Most of the time, the person would get a very mild infection and then be protected. So it was a way of inducing immunity. And it mostly worked, but it didn't always work. And sometimes people got a full-blown case of smallpox and they could risk dying from that infection. So it was better than nothing, but it was still a risky practice. And they were using it because there was nothing better. But in 1796, you might have heard of this man, Edward Jenner. He figured out that if you used a related virus, cowpox, and you gave that to somebody, they would then be protected against smallpox. And cowpox was not a deadly disease. You would get a very mild, set of lesions and you'd heal. So that was the first vaccine. And since then, we've eliminated smallpox from the face of the earth because of the practice of vaccination. So hopefully we'll be able to eliminate other infectious diseases the same way. But so far, smallpox is the only one that has been completely eliminated. So I just wanted to show you this slide. Whoops, jumped ahead. Show you this slide to again emphasize the point that vaccination works. This is just two graphs for two different vaccines against two different infectious diseases in the United States. And you can see, like up on this top graph, when polio was prevalent, there was a lot of cases, right? 40,000 plus cases a year. And polio is a very devastating disease. You may end up paralyzed from that infection. But once the vaccines for polio were introduced, you could see that the case level dropped to nothing. So vaccines were responsible for eliminating polio as well as measles. Measles is shown on the bottom. So when the measles vaccine was introduced, that also caused a severe tapering, almost to zero. We know that some people are now resistant, they don't want to, they're hesitant to get a vaccine. And measles is one of those vaccines that people are sometimes refusing to take nowadays. So we do see little small outbreaks of measles that happens now and again because people are not vaccinated against measles. But you can see that the effect of the vaccine is real. When people get vaccinated, the disease does pretty much disappear from the population. So this slide is a bit complicated, and it's actually more complicated than even this cartoon version. And so I'm not going to say too much, but what I want to introduce to you is the idea that our immune system is pretty complicated, but there's two major branches to the immune system. One branch produces antibodies for us. You may have heard of those, right? Those are proteins made by specific types of cells, and those proteins are protective. And for many vaccines, antibodies are what we are responding with, right? Our body responds by making antibodies that will protect us. You make antibodies because antigens, the things that our immune system is responding to, are floating around outside cells. So those are external, right? They're outside of cells and they go through this complex pathway, and that turns on our immune system to make antibodies. So external antigens give us antibodies. The other branch is driven by or orchestrated by what's called killer T cells. So killer T cells, they're the ones that kill cells that are themselves infected, or if they're altered in some way. So like cancer cells, for example, would be eliminated by our killer T cells. They're the ones that go around our system and look for cells that are changed and not acting normally. And how they recognize that they're changed and not acting normally is that something from the inside of the cell gets presented to on the outside of the cell that's not normal. It's not a self antigen. And the killer cells see that and they're like, you know what? You're not behaving, right? You're not acting like you should. You're infected or you're somehow changed. And they would kill that cell to eliminate the problem. So we have these two branches and they have different roles, and they're both important in their own way. We need antibodies to clear out things that are floating around outside our cells, and we need killer T cells to kill cells and eliminate them from our system when they're not behaving properly. So when we talk a little bit about some of these vaccines, keep in mind that we have these two different branches of our immune system that are working in order to help protect us from developing a disease. So just reiterating of that, the B cells make the antibodies, the proteins. And so these little red Y-shaped molecules are the antibodies. And this is showing some of the different ways that antibodies act in order to help protect us against those externally floating around antigens. They bind to those antigens and they do things to eliminate the threat. So the top is saying, okay, we're neutralizing the threat. So by binding the surface, they can't do whatever it was they were going to do. And this term opsonization means that something like a white blood cell that is responsible for what's called phagocytosis, that's that eating and digesting of foreign material. They help the cells do that. That's what that word means. Agglutination means you're going to stick them all together. So if you clump it all together, they can't move around your body and cause a problem. So again, you stick them all together, you can wait for something like a white blood cell to come around and eat them and digest them and get rid of them. So that clumping is helpful. And then the last one is you have a cell that recognizes when those antibodies are bound, and they come around and they kill cells that have antibodies bound to them. So they kill those cells. So that's all different ways that antibodies are helpful, and that's why we want them. when we get vaccinated or when we're infected and we're making antibodies to clear the infection. This is why. This is the types of good things that those antibodies are doing for us. And this is just showing how a killer cell interacts with that abnormal cell or that infected cell, right? So the infected cell is showing the T cell that it has this antigen, this little yellow molecule that is not a normal self molecule. It might be a viral molecule, or it might be some cancer type of molecule, something that's not normal. And the killer T cell sees it, and that cell now kills the infected cell. So this is how our antibodies and our killer T cells are interacting in order to help protect us. So what kinds of activities do we want to do? So active immunization means we're going to train our immune system, right? We want our immune system to learn how to respond so that we can be protected. So that's what active immunization means. We want our immune system to do it. And the first way we can do it is with what's called an attenuated live vaccine. Attenuated means it's weakened. So obviously we don't want to give somebody a pathogen and make them sick and then make them recover from that. That's defeating the purpose. So what you would do is you can weaken a virus. So for example, the oral polio vaccine is a weakened virus. The chickenpox vaccine is a weakened virus. So the virus can't make you sick, or it shouldn't make you sick if you're a normal, healthy person. If you have a immunocompromised state, that's different, that you don't want to have one of these vaccines. But if you're a normal, healthy person, then when you get this weakened vaccine, you get a very mild infection, but your immune system reacts very strongly. And so you're going to get a strong immune response. The cell-mediated part means your killer T cells will react to this. So that's good, because our killer T cells are going to protect us if that virus gets inside the cell. If we see the real virus, right, the real pathogen in the future, our killer T cells will be able to eliminate those cells really fast. The other interesting thing is that these vaccines can actually provide what's called contact immunity, which is good in limited doses. So if a lot of people are getting the vaccine, but a few people don't get it, then the people that got the vaccine, they're experiencing a very mild infection. They can actually pass the vaccine strain to their contacts. So somebody else in their household, for example. And so in that manner, you can kind of spread the wealth, right? More people can actually be vaccinated than you actually gave the vaccine to. But there can actually be a problem with that if not enough people get the vaccine. There's a problem right now in the Philippines where they were using the attenuated polio vaccine, but so few people were actually getting vaccinated that this contact immunity was happening over and over and over and over again. And the virus actually mutated and started causing disease again. So the answer to that is you have to vaccinate more people with the actual vaccine so that they can be protected. Otherwise, you could have a problem. So there are some issues with using attenuated live vaccines that you have to consider and think about these things so that you're using them in the right conditions. Otherwise, you might have problems, right? Some people are susceptible. If you don't have a good immune system, then you don't want to get this vaccine. And sometimes you have a risk of reverting back to a form that can actually cause disease. So these vaccines have to be used with some caution. They're very, very good. They have very good benefits, but you do need to consider the situation that you're in. The other type of vaccine that's very common is an inactivated or killed vaccine. So a killed vaccine has no risk of acquiring the infection, right? Because it's basically a dead thing. So it's safer. So the whole unit means it's the whole agent, like the whole thing was just killed off. And now you're using that as a way to stimulate your immune system, or you can use parts of it, right? That's a subunit. So just a part of that particular pathogen could be used as a vaccine. They're very safe. You can't get infected by these, but because you're using just a dead thing, right? You get less stimulation of your immune response. So that means that you usually have to have boosters. You need multiple doses. And sometimes it's a little hard, right? Not the whole population is going to be very good about coming back and getting their booster shot. So they might only be partially vaccinated. Sometimes you need other chemicals added to it to increase the ability of your immune response to actually do a good job to respond. So these things are what you have to keep in mind when you are considering what type of vaccine do you want to develop for this particular disease that's affecting a particular population, for example. And then another type of vaccine is a toxoid vaccine. So this is really just one part of a pathogen. It's just the toxin part, but of course you can't give a toxin to somebody because that will be bad, right? It's toxic. It's going to cause bad effects. So usually the toxin is treated with heat or a chemical so that it's no longer going to have the toxic effect, but it still could stimulate antibodies, right? But you probably are going to need those chemical adjuvants to help stimulate your immune response, and you're going to need boosters in order to get a high enough immune response to actually work against the future pathogen that you might come in contact with. So for all of these, for the killed, the subunit, and the toxoid type vaccines, you will only get antibodies. You can't get a cell mediated, you can't get the killer T cells stimulated. The killer T cells are only really stimulated if something is inside the cell to turn them on, right? To activate them. So at this point, only the attenuated vaccines were capable of giving you a cytotoxic T cell, a killer T cell activation. So that leads us to some of the newer versions of vaccines. And all of them use genetic engineering, right? The tools of biotechnology in order to make vaccines. And of course, the goals for all of these vaccines is to make them very effective, right? You want to make them as cheaply as possible, and you want them to be very safe. So those are the goals. So these panels on this slide are showing some of the ways that genetic engineering is used to develop a vaccine. So the first is basically making that attenuated type of pathogen. So you have a living pathogen. It looks like a virus in this case, and they take out a gene. So by taking out the gene, you can't get a full-blown infection, right? A full-blown disease, but you'll get a very mild infection, and that way your immune system can respond. And the idea is that if you respond to this changed, attenuated vaccine strain, you'll be protected against the virulent strain in the future. So that's one, right? We had to take a gene out. The second one is to move a gene into a laboratory cell. So we found an important immune system stimulating gene. You take that out, that's shown in green. So this is a gene that we want our immune system to respond to because it will be protective if we respond to it. We put that gene into a different cell. It could be something like E. coli or a tissue culture cell. And that cell starts making the product, right, of that gene. So making proteins. And then you purify that, and that's going to be part of your vaccine. This other one is making kind of a new combo, kind of a new combination organism. So you take a gene from the pathogen that you want to protect against, and you put it into something that's pretty harmless. So maybe something like a mild cold virus, which is something like the Johnson & Johnson and the AstraZeneca versions of the COVID vaccine that's out right now. They've done something like this, right? They take a gene from the COVID-19, the SARS-CoV-2 virus. They stick it into this other virus that is not really able to cause a very strong infection, but a pretty mild one. And then they give you that. and then your immune system responds. So now you have this recombinant, right? This genetically engineered new combination virus that is a combination of the pathogen, just the genes that you want your immune system to respond to, put into this other virus that doesn't cause a serious infection. And then lastly, we are going to make either DNA or RNA as part of the vaccine. So again, you take the gene that you need your immune system to learn to respond to, you put it into what's called a vector, so a piece of DNA, and then you can put that into E. coli and make many, many copies of it. And either the DNA can be a vaccine, and so far we haven't actually had any of those used widespread yet, but we've had the DNA be transferred into messenger RNA, and then that has been used as a vaccine because that can go inside our own cells, and then our own cells will make that protein encoded by that green gene right there, and then we respond. So that's kind of how the Moderna and the Pfizer vaccines are working. So these are some general scenarios of how genetic engineering is helping us make new vaccines, but we can also look at the COVID-19 vaccine itself and consider some of the different ones that are out released to the public right now. And I'm not familiar with all of the ones in all of the different countries, but I do know several of them that are being used in the United States. So I'll talk a little bit about those. So the SARS-CoV-2 is the virus that causes COVID-19. And we only want to take some genes, specifically the ones from that encode this protein on the surface, out. And so that gene, this virus is actually an RNA virus. And in order to work in the lab, we need DNA. So we have to use an enzyme called reverse transcriptase. That's a tool of genetic engineering. And we turn the RNA to DNA and we amplify it up in a process called PCR. So we make many, many copies of that gene. Then we're going to put it into a DNA vector, so something that E. coli can replicate. So we have to cut it open with enzymes. We use the same enzymes to clip the edges of those PCR products, those genes, and then we glue it into the vector with another enzyme called ligase. So now we have this recombinant vector that has the gene of interest in it. We can use a process to introduce that into a cell like E. coli. E. coli will make many, many copies of that plasmid for us. And so we have a lot of copies of that gene. And so we're going to jump over here for a minute. So sometimes the plasmid itself with the gene could be used as a vaccine, but I should say that as of yet, none of just the DNA version has been released to use for the public, but we can take the DNA and make messenger RNA. If you've had a biology class, you might remember how that process works. So we have DNA being transcribed to messenger RNA. Now the messenger RNA has been used as a vaccine. The Moderna and the Pfizer vaccines are using the messenger RNA, and that gets into our cells. Our cells now make the protein for the viral spike protein, and our killer T cells can respond. So we're going to get antibodies and killer T cells, which is ideal when responding to a viral pathogen, because now both of our branches of immune system are working as they should against a virus. So the messenger RNA can be used, or in the classic types of vaccines, we can use translation, you get the purified protein, and now we could have a subunit vaccine. So right now, I don't know of any subunit vaccines that have been used. The Johnson & Johnson and the AstraZeneca vaccine, as I understand it, are introducing this gene into a different virus, a very weak virus, and using that virus as a vaccine. So this is the different approaches that have been used in order to develop good vaccines. So is there a chat? I think something just popped up. So Hi. So I was wondering if it's possible for someone who took both shots of the vaccine against COVID-19 to not develop immune response. So yes, it's possible. You're saying that, is it possible you got your first shot and you got your booster, yet you get no immune response? And that does happen, unfortunately, sometimes, right? We're going to get to the next topic in a minute. So not everybody's immune system is exactly the same. And so while the vast majority of people will respond appropriately, there's always going to be a small percentage that don't respond well to a vaccine, just because that's a characteristic of their particular immune system. So yeah, it's possible. It's not likely, but it is possible. So let me see. Okay, so actually that's a good question, because it leads to this idea of herd immunity, which you might have been hearing about in the news. This is another chat. Sorry, it's a very, okay, good, thank you. It's hard for me to look at both. Okay, so herd immunity is illustrated in these panels. Let me explain a little bit. So this panel up here with mostly blue people represents something like what happened pre-pandemic, right? Pre-COVID-19, where everybody is not, nobody's immune, nobody's vaccinated, nobody has had the disease and recovered really. So everybody's still susceptible. And now we have individuals that have the virus entering into the population, and it will spread very rapidly. And so that over here on the upper right, you can see that the virus just spreads extremely fast, or the infection can spread very fast, and lots of people get sick, right? So that's what we don't want, really. It's not desirable when we're facing something like COVID-19. What we would prefer is something like the bottom, which is that herd immunity concept. So the people in yellow are now immune. They've either been vaccinated or they had the infection and they recovered from it. So they are not going to get reinfected again. And that leaves fewer susceptible people. So in the blue are the susceptible people, the people that were not vaccinated, never had the infection, or maybe they were vaccinated, just like you asked, and they just didn't respond very well. So they're still susceptible to the infection. So when we have a situation where most people are protected and fewer people are not protected, when somebody that's infected enters into that population, it does not spread very well, right? So if you can see here, over on the right, only one additional person now got infected, and the other people were basically protected because they were surrounded by the herd of people that were protected around them. So the infection did not spread. So when you're faced with vulnerable populations, right, you want to have a good herd immunity. So when you consider something like a vaccine, even though you might think, well, I'm not really much at risk for getting infected, right? Or getting very sick from the infection. So maybe I don't need the vaccine. You might want to think a little bit about that, because if you get the infection or an infection and you spread it to somebody else, right? We're looking more like the situation up at the top. You're going to reach more susceptible people that are at risk of very serious infections and potentially death. So the more people that are protected, the less of that spread and less people that are really susceptible that can die. So we want that herd immunity. We want as many people as possible to be protected so that we can protect as a herd the people that are still susceptible. Okay, so that's that idea. I see another chat come up. Okay, so the difference of producing Coronavac and AstraZeneca with side effects. I'm not familiar with Coronavac, and the AstraZeneca, I believe, is similar to the Johnson & Johnson. So in general, just side effects for normal people for any of these vaccines is what they considered mild. So you might not feel well the next day. So some people feel almost nothing, maybe a little sore arm. And some people get a little sick, like they don't feel very well the next day for all of these vaccines. they feel like maybe they're coming down with the flu or something, it's usually pretty short-lived, 1 to maybe a max of a couple days, and then it goes away. And obviously you're not sick, it's just part of your immune system responding to that. So that's the general side effect. There's, with anything, right? With any drug, with any supplement, with any medication, there's always a low risk that somebody's going to have a bad reaction, like a allergic reaction to it that can be serious, or they potentially could have some other effect, like for a brief moment they paused the Johnson & Johnson immunization in the United States because they were concerned that maybe this blood clotting disorder was related. They decided that it didn't look like it was really strongly related, and it's a very, very low risk, like 1 in a million people. So they re-released it with kind of a warning to look out for some of these symptoms. So that's about all I can tell you about the side effects and the issues that I know of the current vaccines. I'm sorry, I'm not very familiar with the CoronaVac. That might be something that is released in your country, but not ours. Okay. Okay, so I'm going to kind of close out this section on immunization with a word about passive immunotherapy, because you may have heard a little bit about that particular method in treating people that have disease, especially COVID-19. This has happened. So what they do is they give you preformed antibodies, so from somebody else. Now, I don't know if you remember, so Brazil probably didn't have as much about this in the news as the United States did, but back when then President Trump got COVID-19, he got treated, and he got an experimental treatment, and part of that was actually with a preformed antibodies that they made in the lab against the COVID-19. So that's the passive immunotherapy. You get immediate protection, right? So this is, immediate. You give them the infusion, right? You introduce the antibodies to their system, and then immediately the person is experiencing the protection. But what are the limitations? Sometimes you get an allergic reaction with that, especially if you use it more than once, you can get allergic reactions. And then also, if you're isolating this from other people, because that was also something that they were doing for COVID patients, people that recovered from COVID made their own antibodies. They would take that, their blood, right? They donate the blood. They take the antibodies from the people that recovered, and they would give it to somebody that was very sick. And it did work sometimes. It depends on how sick the person was. If they were very, very sick, it might be too late, but it was helping. So that's passive immunotherapy, but they degrade, right, very, very fast. So if you're taking it from a person, there could be other viruses that in there. What if the person had hepatitis or, HIV or something? Sometimes those can travel along with the antibodies that you're trying to give them if you don't screen for them specifically and try to make sure they're not there. And they don't last very long. This is a very short-lived thing. Immediate protection, but it's going to go away. You're not going to have this effect of being protected for very long. Is there another chat?
Yeah, we have a couple questions in the chat over here.
So if I don't spread the virus dies. Yeah, basically. So yeah, that's the good thing about that's why people are quarantining, right? If your immune system clears out the virus and you don't spread it to somebody else, that you're the dead end, right? The virus doesn't go anywhere else. So that's a good thing. That's why they don't want you to spread it. And then the other question, so the side effects don't have relation with the way they produce the vaccines. It depends, right? So the different types of vaccines might have slightly different types of side effects, but they're usually pretty mild. Hopefully that answers that question. Oops. Okay, so almost at the very end. So I just wanted to compare the active immunization, so you're giving, you're training your own immune system. That's the active immunization versus that passive where you're giving somebody antibodies that were made somewhere else, right? Not in their own system. That's called passive immunotherapy. So in the red line, you can see that you give someone a vaccine, right? The initial inoculation, their immune system starts to ramp up production of antibodies. And then these arrows are showing booster doses. So you get, maybe one booster and maybe later another booster. And you can see that your antibody concentration in your system is going to go up, 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 up. And this is long-term. They're going to stick around. You've trained your immune system to recognize it. And now you're going to be able to respond yourself. Whereas in this kind of greenish blue line, what happens is if you give somebody preformed antibodies, right, made somewhere else, either in another person or made in the laboratory, and you give them those antibodies, they'll initially have a very high concentration in their system, which hopefully will protect them, help them clear out that infection. But over a fairly short period of time, that concentration is going to just drop off because your system is going to clear that out, and you do not know how to make them yourself. Your system has not been trained, so those will disappear. So those are the two methods of protecting somebody against an infectious disease. And ideally, we give something like a vaccine to train your system so that you yourself will be protected. We don't really want to just give everybody a bunch of passive protection because it's so short-lived, and you might have to do this over and over again. And especially after the first dose, your risk of having something like an allergy is very high. And it's also very, very expensive to do that. It's much less expensive to give an initial vaccine. Okay, so before I move to the scenario, let's see if there's one more. Okay, thank you. All right, so here's my little challenge to you all for a few moments. So you're a researcher, right? There's a new viral pathogen that's been discovered, and it's causing a lethal infection only in young children. And it appears to be related to measles. It attaches to cells with a specific protein. It's out on the envelope on the outside, and it initiates infection that way. And other viral proteins help replicate the virus inside host cells. So if you're a researcher that wants to quickly develop a vaccine, what kind of vaccine do you want to investigate? Think about the different types that we talked about and why. And would you rather have an antibody response or a killer T cell response against this particular type of pathogen? So let me get the chat. Okay, somebody says sarampo. So that's what you call it. So someone put some ideas out. You can either use your mic or type your ideas in the chat and we'll talk about it. So what are the types of professions that work with vaccines? So you could be a researcher that's developing a vaccine, right? Or investigating that. And that might happen at a place like a university lab, or it might be happening in a big company lab. Think, Johnson & Johnson and Pfizer and Moderna and BioNTech, right? So it could be a company that is employing researchers to investigate vaccines, or it might be happening at a university lab where somebody is looking at that. So those are different types of professions that are investigating the initial vaccines. And then of course you have the companies that are producing them. So there's all kinds of biomanufacturing jobs where you are going to be producing components for the vaccine or packaging the vaccine, right? Somebody has to put the vaccines in the little vials and they have to be sterile, and you have to ensure the quality of those vaccines and make sure that they are working correctly each and every batch. So you have the companies that are making it and manufacturing it, the companies that are researching it, or you have research in laboratories. Does that help? What about chickenpox? I don't understand the question about chickenpox. Can you elaborate a little bit? So, okay. Could I use the chickenpox virus to create the antigen? Oh, attenuated vaccine. That's actually a good idea. Okay. So similar to the chickenpox vaccine, to an attenuated vaccine, that actually is probably a pretty good approach, right? The chickenpox vaccine is given to children. So we know that that one must be safe. So maybe you're a researcher and you use that base attenuated virus of the chickenpox and add some new genes to it from this new virus, and you could maybe potentially even protect against chickenpox and this new virus, right? A new combination vaccine. That actually could be a really good approach. So yeah, good idea. That would be definitely something that you could research and see if that worked. So I saw something about antibody response. So antibody response will happen, right, with pretty much any stimulation of your immune system. But if you use the attenuated vaccine, remember, you're able to stimulate your cytotoxic T cells. And for a virus, that's actually something very beneficial because viruses go inside our cells, right? And so, in order to really protect us really well against a viral infection, it would be good to have an antibody and a cytotoxic T cell response, not just antibody. So, attenuated has the benefit of allowing us, if we're using an attenuated viral vaccine, we're going to be able to get a cytotoxic T cell. That's really a good thing. Okay, because it's similar to measles, we know this pathogen. Yeah, you probably do know some things about the pathogen. So the herd immunity and the effectiveness of vaccines, how does the effectiveness affect herd immunity? Okay, that's a pretty good question. So if the vaccine is really good, right? Like some of the vaccines to COVID-19 look like they give a really good protection. That means that we can generate herd immunity a little bit more efficiently because there's going to be less of those people that get a vaccine and aren't protected. So the better the vaccine, the faster we can get to herd immunity as people get vaccinated. So we want more people to get vaccinated because we'll get there pretty fast. So how does the anti-vaccine, how does it, I don't know about I don't understand the anti-vaccine movement. It's just a phrase. So what's the question about the anti-vaccine movement, how it affects it? The fact that people are worried, okay, affect the herd immunity. Okay, so that actually is what happened or is happening with measles. We have a history of seeing that in, for example, the United States. So people are more afraid of the vaccine against measles than they are of the infection itself, because at least here in the United States, we've sort of forgotten how bad measles can actually be and what the long-term consequences of getting and recovering from measles is. So when people don't take the vaccine and an infected individual enters that little population, and measles will spread very fast. So, the problem with people being very hesitant to get the vaccine, being very scared that the vaccine is worse than the disease, is that we don't get herd immunity fast, and there'll be pockets of the population that are going to be super susceptible, and it's going to spread. So yeah, the anti-vaccine movement does affect herd immunity. It decreases our ability to get there. So it's better when people investigate the science and look at it more critically instead of just like listening to what happens in the popular news sometimes. that isn't always very accurate from a scientific point of view. And then, people get scared. It's pretty easy to scare people. And it's a lot harder to convince somebody that's now scared that it's okay. So once you've scared somebody, it's hard to convince them that it's safe to do something. So it's better to be upfront and have people investigate the science first and really look at it critically before you get a lot of negative, anti-vaccine things out there. So antibody response sounds good. Antibody response is always good. Antibodies do protect us against all kinds of things. And as long as your antibody response is strong enough, you will be protected against a virus as well. But, excuse me, when viruses get inside our cells, antibodies no longer work because antibodies only work outside cells. And a virus, once it gets in, is hidden from the effects of antibodies. And that's where we would need cytotoxic T cells. We need those killer T cells to come in and kill an infected cell. So ideally, for a viral pathogen, we want antibodies and T cells, killer T cells. Are there other questions?
I have two, Professor Denise.
Sure.
So why there are different infectious disease that we still don't have a vaccine, like HIV in Brazil, dengue as well?
Mhm.
And the second one is why you said that AstraZeneca and Johnson vaccines are similar, but one is 2 doses and the another one is just a single dose. So that's my questions.
Okay, so let me answer the second one first. So sometimes, especially in this climate where vaccines are being like approved under emergency situations, they're using whatever protocol the company tested as the way to go, right? So if a company gave 1 dose and then they checked, they went through all the trials and the clinical trials and the safety trials, and they used just 1 dose, then that's what they're recommending to be used at this point. If the company used more than 1 because they weren't happy maybe with the enough of a response the first time, or they assumed you would need a booster, and so they did 2, then that's what they're using. So in this particular moment, the exact approach that they're using might not be ideal. So for example, it's not that it's unsafe, it's just that they just are using what they know is going to be effective under the situations that they tested. So for example, for Pfizer at first here in the United States, it had to be transported at -70 degrees Celsius. That's extremely cold, and it requires a special freezer in order to do that. It's not like the freezer in your home, right? But that's because that's how they tested the virus, right? That's a laboratory freezer. A lot of us have -80 or -70 freezers in our labs. So they were working with that, and that's the data that they use. So that's exactly how they had to distribute it. But as soon as they got approval, they started they tested, right? Can it be stable at a little bit lower freezer temperature, something more like in our house? And so, yes, they found out that, yeah, you actually could just put it in a regular freezer. And so then kind of a modification went out to that protocol saying that they could use it. So what I'm saying is that while these vaccines, the AstraZeneca and Johnson & Johnson, may be similar, they're just using whatever the company said, because that's how they tested it, because everything was tested very quickly to make, not that the numbers were less or that the clinical trial was less, but that they had to design their experiment and then move forward with it with all these many thousands of people that they were testing to see if it was safe and if it worked. And whatever the data said at the end, whatever their protocol was, is exactly what they're doing. It may be that in a year or two from now, when they have tried a few other things and they have more data, they may modify it, just like they modified the transporting of the Moderna. So I hope that helped answer that question. The thing about why we don't have vaccines against some diseases, it's because it's tricky. I made it sound somewhat easy when I showed you how we can move genes or whatever, but the tricky part sometimes is finding which gene is the one that will make our immune system learn to respond and protect. And for HIV, this is a virus that changes. It's a mutating virus. It changes all the time. So if our immune system sort of learns how to respond to one version, it switches those surface proteins a little bit, and then our immune system can't really recognize it anymore, and it has to relearn. And so if you develop a vaccine against just one strain of HIV, it doesn't protect against anything for very long. So it's a bit tricky for some of these diseases to find the right thing to make our immune system actually learn. And I made it sound kind of easy, but there's a lot of research that goes into these things to find. And we were in some ways lucky that the COVID-19 disease had a gene that we could use that they knew very quickly, right? They were able to develop a vaccine very quickly, because if it was something like HIV, we would be in a pandemic situation for a very, very long time for them to figure out which vaccine will work. So in some ways, even how as terrible as this pandemic is, we're lucky it isn't worse. It isn't something like HIV where we can't figure out which vaccine might actually be effective. Okay, so there's a question. In the case of viruses like COVID-19, is it possible that vaccines we have up to date obsolete against new variables? Strategies to deal with the mutation rate when you're developing a vaccine. So yes, it's possible that a new variant is going to emerge that is not going to be protected against with this current vaccine. And I think they're already trying to develop, some of these bigger companies, right, are already trying to develop versions of the vaccine that will be better against some of these variants that are developing. That's why they are saying that our best approach is actually to try to vaccinate a bunch of people as fast as possible so that there's fewer of these mutated variants that emerge out of the population. So if you can stop the virus from replicating so much in our population, then you can help slow down the effect of the virus developing a mutated version that will escape. So sometimes it's pretty hard to figure out what the strategy would be to deal with it. You basically are doing what we're doing now, watching what's happening, trying to learn what the virus is doing, and then develop a new version of the vaccine. So the best strategy is to not let the virus spread. So that's the best strategy. It's not easy though. In a hypothetical situation, a senior that gets 2 doses of COVID-19 and gets in contact with contaminated people, can he spread the virus? So you're saying if the person has had their vaccination and then they come in contact with somebody that has COVID-19, can they spread it? They don't know for sure, right? There's a little bit of question right now. They don't have a perfect answer, but it looks like they're not going to spread it much. There is a small risk that they could have the virus multiplying for a short time in the vaccinated person, and during that time they could spread it. But because their immune system, if their immune system has responded well to the vaccine, their immune system should clear out the virus. So low risk, but theoretically, yes, they could for a little bit. So I think I should just kind of close out. I know we're running out of time. You can keep asking questions. I just wanted to talk just really briefly so you'll know a little bit about our program. And I know that you can reach out to your people and our people to get more questions answered. But just to give you a kind of an overview, what our students do, in our program is they can earn certificates. You can get a bachelor's degree in biotech after transferring from our college. At our college, you can get an associate's degree in biotechnology, or you can get certificates. And the first certificate is really only 4 classes. So the first 2 that are listed here are linked. So you have a lecture and a lab, introduction to biotech. Then you have a basic skills class and an intro to chemistry class. And if you finish this first certificate, then you are eligible for getting some help on our campus for landing internships and potentially jobs if you're eligible to be employed in the United States. And then we have a second certificate that builds on the first, and you take a few more classes in biotech, one on proteins, one on the regulations in the industry. So something we were talking about when you manufacture these different components like vaccines, what are the regulations that these companies are dealing with? And then how do you ensure that those products are quality products and are going to do exactly what they say they're going to do? And then you take another chemistry class. And then the last certificate takes a class that's on nucleic acids and also tissue culture. And then you can have some electives that you take. So this is how our structure is set up, where you have different certificates building upon each other. And then you can also earn an AS degree, and there's also a BS degree in biotech. So is there any other questions, or Malka, is there any other things I should cover?
I think you covered all the questions in the chat. Thank you so much for the class today, Dr. Foley. It was amazing, and the questions were so good. Oh, I think we have a last one.
I see one. So someone's asking about killer T cells operate differently than leukocytes. So leukocyte is a general term. It means white blood cell. So there's different varieties of white blood cells. So you can have like macrophages, like I was talking about, the ones that eat up and digest, right? They phagocytose things. The killer T cells are a leukocyte. They're a lymphocyte. So the T cells and the B cells that make antibodies are lymphocytes. So they're part of leukocytes. They're part of that group.
Any more questions, guys?
Thank you very much.
Thank you so much, Dr. Foley. It was amazing class. Pessoal, não esquece que tem aula segunda-feira. O cadastro já está aberto no Simpla, tá bom? Thank you so much, Dr. Foley.
Thank you. Have a very good day, everybody. Hope you enjoyed it. That was a good group of people, huh? And there was a lot of people. Thank you.
It was a lot of questions. That was so good.
Good. Okay. Anytime.
Thank
Happy to do something
you.
again in the future. Thank you very much.
Thank you so much, Dr. Foley. Thank you, girls.
Bye.